All right, so last week we had a nice chat to make sure everybody is up to speed, right? We covered about 300 years in detail, right? So that's pretty impressive, right? So we covered 300 years of neuroscience knowledge last week. So you're definitely getting your money's worth. So I want to make sure that happens. I really have considered, Casey, this is sort of like a Shakespearean break the fourth wall sort of aside between me, the main actor, and you, the audience, right? You didn't know you were getting this like high-level literary concepts either. I don't know if any of you guys have had to study Shakespeare. It's largely a waste of time. I mean, it is largely a waste of time. Chapter 2 is probably the most difficult chapter in the entire book. It's long, it's detailed, it's very, for like one of you, you're going to get excited, JP, right? Because it's very molecular, it's very, we're going to get down and talk about ion channels and sodium and all kinds of fun things like that. It's going to be awesome. But I've actually toyed with like trying to move this after the drop ad date, right? So because this is a difficult chapter, okay? You just got to hang with it, though. I promise it's going to be helpful. Once you get your mind wrapped around this, it's not going to be so bad, right? There's going to be a lot of info on these slides. We're going to skip some of it you may already know. If you need to go back to it, it's going to be there. We're going to hit the important parts, okay? So that's going to be awesome. We'll hit what's important for you. We're going to dwell on it when we need to. We will constantly be calling back to this stuff, okay? So hang in there. We're going to keep going back and back and back, okay? So it's going to be great. But we are going to talk about structure and function of cells of the nervous system, right? So we'll talk about cells of the nervous system, communication within the neuron, right? Like how do disparate parts of the neuron communicate within itself? How do we pass that signal around? And then what's really important is like, you know, just because if you just sit around and talk to yourself all the time, Morgan, what does that do for you? Well, that probably gets you some funny stares, right? But it doesn't really move you along in the world, right? You need to be able to communicate with other entities, right? So we're going to talk about communication between neurons as well. So that's going to be exciting. We're just going to go all the way back and we're going to zoom in, right? We're going to keep doing this all day. Your nervous system can be divided using a variety of criteria, right? Okay. One's not necessarily better than the other in the way that we think about dividing the nervous system. It's going to serve different purposes, right? To think about things differently. Okay. So if you try to think about like, how many of you are baseball fans? All right, we got a few baseball fans. So you might want to divide people by position, right? So you can think about a shortstop versus a third baseman, maybe. That might be helpful. Another way to divide players is simply by, pitchers and batters, right? Or left-handed batters versus right-handed batters, right? Those are not always, you got to use what kind of division makes the most sense for what you're trying to accomplish. We're going to do that with the nervous system. The first sort of basic way to divide this is your central nervous system, or CNS, consists only of your brain and your spinal cord. That's it. Brain, spinal cord, central nervous system. Anything sticking off the brain or off the spinal cord, right, that's going to be part of your peripheral nervous system. Okay? There are important distinctions between the central nervous system and the peripheral nervous system aside from their location, right? We'll talk about those when we need to, okay? But first we just want to think about location, central versus peripheral. Central nervous system, we can think about this. It's always encased in bone, right? How many of you have a skull? Okay, the rest of you, that's what I thought. So, you've got the skull and your vertebral column, right? Okay, that's the bone that's going to encase the brain and the spinal cord. Other stuff's going to come out of the holes of those bones, right? Those holes have fancy names. I'm not going to ask you to know all of them. probably 95%. No, you're not going to have to know any of those really. You're going to be fine. But stuff coming out of those holes into other parts of your body, that's going to be your peripheral nervous system. Central versus peripheral. Here's a little preview of a good, important difference. Damage to the central nervous system is typically permanent. Okay? There's not much recovery from damage to central nervous system. Peripheral nervous system damage, there can be some recovery. And we'll talk in detail in a moment. So I want you to keep this CNS, PNS division in mind when we talk in just a few moments about oligodendrocytes. Okay? Because there are two kinds of cells that create the myelin sheath, some in the central, some in the peripheral, and they have some important differences. Okay, so it's going to highlight why this difference is important. Just in case you decided not to listen to anything I said and you prefer colors, there's the diagram. And I'm just going to draw green right around all of this so you know it's covered in bone. Hey, you know what's awesome? Your spinal cord doesn't go all the way down. That's cool. When you get down to the bottom, there's a thing called the cauda equina. It means horse's tail, so like all of the other like stuff from like here down, right? So it just hangs there. You also have a thing called the filum terminale. This is what keeps your spinal cord from rolling up into your brain. Okay, so you got to watch that. Okay, no, huh?
You don't want to sever that.
No, you don't, because then, like in other things, can shear, right? And then you've kind of lost it all, literally. So, so be careful. Like I always think when people leave this class, you're gonna walk like this, like this. Don't want to disturb it. Just keep it intact. Okay, but if you think about all the idiot things you've done so far in your life and you've not damaged it, you're probably okay. Okay, trust me. All right, that's about all you need to know there. We like to think about sort of three things in the central or in the nervous system. We like to think about sensory, motor, and interneurons. Not in that order. I don't know why they tell them to always in that order, but that's the wrong order. It should be sensory, interneurons, and then motor neurons probably. I think that's okay. But I like to think about a different sort of way of thinking about your nervous system, and I love this application because this is not in your book, by the way. So this is something you really need to listen to, Casey. It's going to change the way you view your world. Your nervous system does three things. And your whole nervous system does this, and this works all the way down, right? It doesn't matter. There are only three things that happen. Okay? That's it. It's input, calculation, and output. That's all there is to it. Okay? We can think about it in terms of sensory, interneurons, and motor neurons if you want. That's fine. They map onto that at that level. If you want to zoom out from that a bit more, it's going to map onto your whole system, right? What is the purpose of your nervous system? To collect data, do some calculation with that data so you can make some kind of action, right? Some kind of decision, some kind of output. If we want to go all the way down to an ion channel, what does that ion channel do? It gets some sort of input. We'll talk about what that is. It, based on that input, makes some sort of change, and then there's some action, some output, right? So this input, calculation, output, you can apply it all the way across the nervous system. Whether you're talking about a single cell, you're talking about a group of cells in a nucleus, you're talking about a circuit, you're talking about an entire, let's say your entire visual system, right, which is going to encompass a lot of circuits, or we're talking about your entire nervous system for the entire organism. Input, calculation, output, okay? That's really how I want you to think about this. That's all that's happening at every single level. This is a nice, broad, overarching theme. It's pretty good, right? Hey, what's this? This is a neuron. It's a generic neuron. Much like that blobby catfish cell thing that they showed you in biology class, this thing probably doesn't exist, at least not in this conformation, right? But it's good for illustrative purposes. Let's sort of show you what's going on. So what are the basic parts of a neuron? Again, keeping in mind, what does this neuron need to do? Three things. Input, calculation, output. That's it, right? Input typically is going to occur out here at your dendrites. Please do not say dendritic tree. That bothers me, Casey. So you had this face like I've said that before.
I've never said that.
Great. But you know people who have. Some people will say that. It's okay. It's like saying tree, because dendrite is tree, right? If you say like dendritic arbor, I'll kind of let you buy with that, right? Some people will say dendritic arbor just to sound fancy, right? I hate when people do that. They're dendrites. It's from the Greek word for tree. Obviously, it looks like tree branches, right? You saw that, right, Chiquilla? You knew that. Whatever. These are your dendrites. They are the primary input area, right? And these dendrites are underrepresented by a factor of about an infinity, okay? Because they're just all over the place. I'm going to draw in all the rest of the dendrites for you. Okay.
You missed something on the side there.
Thanks. All right. Now let's just get those off there so we can actually see what's going on. This is a drastic underrepresentation of your dendrites, and that's fine. You got a bunch of these. These guys are responsible for catching, on average, 10,000 other inputs, okay, from other cells, right, from other neurons. Some of these will only collect one input. That's a very special case in your retina. We can talk about that. Some of them will collect as many as 100,000 inputs. Remember that guy Purkinje we talked about? You've got Purkinje cells in your cerebellum responsible for coordination, right? Those guys will collect like 100,000 inputs. Okay, that's actually pretty impressive. They happen a lot on these things called dendritic spines. These little spines will come off. They increase surface area. Surface area to volume ratio is very, very important, okay? And this is an important concept just for your own understanding of life. You should sort of have this, right? So, Jawan, what's your favorite cell? And the answer is, if you can't say neuron because you're not going to be able to, red blood cell. That's a cool one, right? Okay. The reason people love red blood cells, it's also shaped like most people's favorite food. It's a donut, right? How many of you don't like donuts? Nobody. Nobody does not like that, right, Casey? I mean, you may not eat donuts for different reasons, but you still like donuts. Given the option between donut or no donut, you always go donut. Okay. Red blood cells are shaped this way because it maximizes surface area while minimizing volume, right? That's awesome because you want oxygen to diffuse in and out, right? Okay, so that's what's going to happen. Plus, you can jam it down in those capillaries. We're doing the same thing out here in the dendrites by adding all of these little spines. We can drastically increase the surface area, right, without increasing volume. How many of you have ever looked at that funny striped thing in the front of your car when you lift your hood, called a radiator? Yeah. Why does it have all those fins? Surface area without increasing volume. If you were to make that surface area and not have those little fins, your car would it'd be massive, right? It's the same thing with your cortex. Why is it all wrinkled up? Okay? Not to confuse you, to fit more in there, because it's really like a square meter, right? And can you imagine, remember that whole conversation we had, and you all called your mom and apologized for how big your head was, right? And we talked about how big would your head have to be if your cortex was smoothed out? It'd be like this big. And that's just ridiculous. Surface area to volume. That's what we're doing there. We're maximizing surface area so we can collect those 10,000 inputs without having this massive cell, right? Because we want to cram like trillions of these guys in something like this big. Because that's how big your brain is. Believe it or not. I know you all think like, man. So take your head, and then first you got to cut off your face. And not just the surface of your face, but the back of your face, right? So you got to take a big chunk out of there. Because a lot of you have big mouths. Then you've got to take off like hair. Hair it'll fool people, make people think their head's bigger, right? If you got a lot of hair. Take off the hair, take off the scalp, take off the bone. Then you got some other space in there we're going to talk about. I mean, you get down. It's really like a grapefruit with a pear on the back. That's about what you got up there, right? They're super small. And your spinal cord is smaller than your pinky. which is frightening. I mean, it is super small, and you look at that and you think, all of the information that my body collects is coming through that thing, and then all the commands going back out. It's amazing. So, we've got our dendrites collecting all of that information. That information, we're going to talk more about this later. It's going to be electrical signals, right? In the way of, it's going to be ion movement in and out of this cell. We'll talk about all that. It's going to be exciting, I promise. I want to go like electrifying just to make some kind of extra pun on top of this, but I'm going to spare you guys that. Okay. Then I want to like make another like higher level joke about capacity, but you're not going to get that because you don't understand capacitance. But if you did, boy, it'd be a great one. So all these we're going to collect. We're going to collect signals, yes notes, yes votes, no votes, right? Constantly up and down. Okay. So we're going to be getting a lot of ion channels that are going to open. It's going to make this guy more excited, more likely to pass on a signal. Have a bunch of ion channels open. It's going to make this guy less excited to pass on that signal, that action potential, right? We'll talk in more detail in a bit about that, so just hang in there on those fuzzy details. All of that info eventually is going to flow down to something called the soma, or cell body, okay? And then it's going to go out through the axon. We'll talk about that in a minute. What goes on in the soma? Again, we're still going to be doing those calculations up and down on these, you know, we'll call it potential right now. You don't know what that means. We'll explain potential in a bit. Potential goes up and down. Eventually, we're going to make that decision to send or not send that signal. Other things that happen in the cell body are the same things that happen in every other cell, right? This guy needs DNA. It needs ribosomes. It needs mitochondria, right? Okay. All of that stuff's going to be going on in the cell body, the soma. Okay. Not a big deal. Right? So we've done our input, we've done our calculation. We've got to think about the output. We've got to try to send this signal somewhere, right? And we'll do that down a guy called the axon. The axon is really just a cable connecting your cell body to your terminal buttons. Now, some people will, again, the same people who say dendritic arbor will all say terminal boutons, right? Boutons. Really fancy it up there. Call it a bouton. If I catch one of you guys doing it, I know you're just trying to impress me. And it's really going to make you look bad. Okay? They're terminals, whatever, I don't care. Call them I don't even call them terminal buttons. That's kind of ridiculous. They're terminals, right? That's what they are. When we get to the muscular system, we'll call them synaptic end plates. Whoop-de-doo, right? It's the same thing. We're just going to throw a different name on it to confuse you. I think that's a waste of time. They're all terminals. They're going to do the same job, okay? Not a big deal. This guy's actually going to connect back on probably to a dendrite of another cell, right? So it's going to create this, not a loop, it's going to create this continuous string. Sometimes it's a loop, right? Kind of depends on what you want happening. We're going to talk a lot about axon mechanics, what happens in that axon, why the signal travels as it does, why it travels fast. We're going to get details later. Let's just sort of build up what we're doing. All along your axon, you've got these sort of, from a cross-section, looks like a jelly roll, right? We're just going across all the snack cakes today. We did donuts, we're going to do jelly rolls now. It's going to look like a jelly roll. Basically, it's a fatty sort of extension. We'll talk about it in a moment of what kind of cell it is. A fatty extension of the cell. It's going to wrap around there, provide some insulation. Okay? Does a couple things. It keeps the signal contained, right? So it's high fidelity when it reaches the end of the axon. It's like, yeah, that's exactly what I meant to send. Perfect. It also travels faster. We call it saltatory conduction. Later, you're going to get confused and think it has to do with salt. It doesn't. It's leaping, right? Also, these gaps are called nodes of Ranvier. If you say Ranvier, I will tell people I don't know you. There you go. Ranvier, that's an I. Do you know someone who said Ranvier?
I've totally said it.
It happens.
You're going to have so many students from my 201 classes come in here, they're going to call.
I fix it.
Okay, good.
I'll tell you why I know. This guy was a French Canadian, Ranvier. A Canadian taught me this. So it's a little, you know, country pride for him. He was not French Canadian, in fact, which was kind of strange. His father was a Western separatist. So I'm not surprised he didn't pronounce it Ranvier on purpose just to insult the French Canadians. But he didn't. He was a very nice guy. I don't think it had anything to do with him being Canadian. There you go. Actually, his son was a baseball pitcher for the Blue Jays briefly and then the Athletics as well. His uncle played for the Blackhawks hockey. So, not that that's kind of a weird blip in the middle there, right? Like an uncle who's an athlete, a son, and then he's like, I'm going to be a neuroscientist. It's like, all right. That's the way it goes, right? So you got your nodes of Ranvier there, speeding up that signal. We're going to get down to our terminals. When we get to our terminals, as I said, we need to pass that signal somewhere else, right? Everything that's going on inside of this cell, we'll talk more about this, is electrical, okay? It's all electrical signals, millivolts, okay? It does not work well, although it happens. Some places we're not going to worry too much about that. In general, it's not a good idea to wire up all of these guys with electrical connections, right? How many of you have a place where you live? That's not a joke. I'm not trying to find out who's like in a housing crisis. I'm just, people live places. And those places have light switches. And they turn on different things, right? Okay, they don't turn on everything. If all of your neurons were connected electrically, every time you turned on one, all the others would turn on. And that would be like walking into your home and turning on your light switch and your washing machine starts. Well, that seems ridiculous, right? Those need to be on different circuits, okay? When you turn on your circuit that moves your right arm, you don't need your left leg to move every time, right? Sometimes you do, sometimes you don't. They need to be separate circuits, okay? So we need to have this disconnect. There are times where we want to electrically couple neurons. That's great when we want to synchronize these signals, right? Those are sort of special case scenarios. Most of you don't need to worry about those, okay? Those of you that do need to worry about it, you'll know. You'll receive a letter in the mail. Questions about this? Great. How many of you are lost? How many of you are so lost you didn't remember to raise your hand when I asked if you were lost? No one? Great. Guess who already talked about all that stuff? We did. Now, this is a little more complicated, right? So we've plugged in a couple extra neurons, showing you some of the other possibilities, not all of the possibilities, just some of the possibilities, right, for how we can connect this. Most of what you see are what we call like dendritic connections. So, you know, we're just connecting onto dendrites. You can connect onto the soma. That's pretty awesome. You can even connect down here at the terminals. Or an awesome place to connect is right here. There's an area there called the axon hillock. Okay? The axon hillock is a really important place, right? I think most of you as individuals don't know where your axon hillock is. And what I mean by that is, how many of you have difficulty making decisions? Right? Yeah, there we go. Hey, thanks for being brave. The rest of you were so bad at making decisions you didn't even know if you should raise your hand. Like, I don't know. Is that me? Maybe. I don't know. The axon hillock's a decision point. Once you get to the axon hillock, guess what's going to happen? Action potential. Right? Now, if you get right up to that axon hillock and you go, backing out, that's fine. Okay? It's sort of like, you know, you're getting ready to run down the aisle, and then you can just the other way if you need to. Right? Once you get up there, believe me, they will make you go through with it. So, just as a warning for those of you, anybody considering marriage, I don't know why I'm writing in red. I'm just kidding. I think it depends on like who's down there, like waiting on you, right? I mean, who said that? Somebody's like, I mean, you know. There you go. Right? So, axon hillock, once you get past that, you're going to fire an action potential. What's a great thing to do right there is put a big old inhibitory synapse, right? Because then what you can do is you can go, nope, don't want to do it. And you can just really put a real stop on that, right? I was thinking about this today. How many of you have ever put gasoline in a vehicle? Not in the tank, but somewhere else. No, nobody. I don't know. So when you're pumping your gasoline, one of the things you should do is always try to juggle flaming bowling pins. No. You can see that. That sounds good, right? One of the things you should do is always look for the emergency stop button, right? On the side of the building, there's an emergency stop button, right? It's a big red button typically. Red usually means whoa, right? So there's a big red button. Most of you didn't know that was there, which I hope none of you are gas station attendants. If like things catch on fire, guess what? Somebody can hit that and it's going to stop the flow of gasoline, so we just not like keep shooting flames. Okay, that's exciting, right? Yeah, fun fact to know. Next time you walk into Sheetz, ask the guy, but be careful how you ask. I feel like there's some things you don't say when you walk into like certain places, and like you don't go, hey, where's your emergency stop button? I don't know. That just seems like that could, especially in this town. It does, right?
It's just a little bit.
Because I learned my lesson once, JP. I went into the bank. This is a good one. I go into the bank and I have like this change that I've rolled, and so I was in graduate school at the time, so you know it was at least $8, right? Because that's about how much money I had, right? It was not a lot. It was like less than $25, I promise. So I go in. It's like 20 minutes until they close, right? So it's like 4:40, whatever. I can do I think if they close at 5, as long as I can get in and finish my business by 5, you're going to handle me, right? If I come in at 20 minutes till 5 and I go, hey, I'm going to apply for a home loan, then you can turn me away and say, come back tomorrow, right? That's fine. You probably won't because it's like a lot of money. But I'm going in, and the teller, she looks at me and she says, I'm going to try my best to mimic her attitude. Is that a bag full of change? And I said, first thing, I didn't have this inhibitory synapse apparently at that time or now. And the first thing that comes out of my mouth is, it's much better that I walk in with a bag of money and say, here it is, than to walk in with an empty bag and say, fill it up. Which apparently is not a joke you tell at the bank. I will tell you. And she said, oh, no, you can't say that. And I was like, well, here's my change. And they counted it quickly and I left. So, I didn't have any more problems at that bank. So there you go. Inhibitory synapses are great. They can shut things down before they happen, right? So you might want to put one of those there. How many of you like making cookies? So we're asking about making first to see if there are enough people here who can bring them so we can eat them. So I saw a few hands go up, so we're fully anticipating cookies next week, right? So do you need to have them raise their hands again, write down their names? Okay.
She's ready, man.
What's great about cookies is, especially if you make them in your kitchen and then you like bring them to wherever I am, right? That's great. And to be able to transport something from one location to another, right? Okay. And we need to do this because look at this thing. How long do you think this is? This is probably like 700 feet long. No, neurons can be quite long though, right? When you think about this, okay, they have to travel, right? Some extended distance, especially given the diameter. It's really a long distance, relatively speaking. And we need to make things in this soma, and we need to get them down here, right? Because most of your sort of cellular machinery is up in that soma, right? So we're going to be making proteins. What are those proteins? Ion channels. We're going to make neurotransmitters. We're going to have to repair things, right? We're going to be sending stuff down. Then there's going to be stuff at the end there in those terminals that it's going to be broken, and we have to send that back for recycling, right? Okay, so we have to have things go both directions, right? Now we call this Axoplasmic Transport. Why? Because it's really a fun name. Axo, we're talking about the axon. Plasmic, think about the cytoplasm in there, right? We're going back and forth on that. Now, we're going to move up and down these things called microtubules. Microtubules are awesome. They give that neuron its structure, right? So you can have that long extended axon and has enough rigidity to stay there. but it also allows you, it's like a railroad track, right? So you can move things on these microtubules. We move things in two directions. There's anterograde, that's soma to terminal. Antero means forward, right? So we're going front, anterior. So we're going forward. Retrograde means moving in reverse. We're going to go from the terminal to the soma for recycling. That's simple. There is a very complicated and fun process I'm not going to worry you too much with, right? I know. You can read it on your own time and get really excited about that. But I don't think for this class that's going to be that thrilling for you. All right, here's sort of the internals of a neuron. We're not going to spend a whole lot of time on this, right? It's basically the same stuff that's in every other cell. You are going to see a high number of mitochondria, right? In neurons, neurons are very active cells, right? So we need those mitochondria. Why? What are mitochondria? Yeah, powerhouse of the cell, right? Everybody knows that. Great job giving us an original answer. I know, right? Somebody goes mitochondria, like powerhouse of the cell. Yeah, they create ATP. Casey, did you have to go through this training? I'm asking you this as a semi-colleague, as a teaching assistant, where you had to like talk about like triggers and things like that, like things you might talk about in class that could like give students bad reactions.
Yeah.
Okay, I'm getting ready to say one.
Okay.
Krebs cycle. That was funny to like the 3 people who know the Krebs cycle, because I'm certain that's giving you a mental disorder trying to learn the Krebs cycle. I won a Nobel Prize for that. The Krebs cycle is a way that you create ATP. We're not going to worry about that. It goes on in the mitochondria, right? So you're okay. Here. Nucleus is important. We are going to talk about DNA. Right? Not today, probably. We'll talk about it some other time. DNA is important, right? Because we'll talk about genetic mutations. We'll talk about the DNA because we're going to create proteins. We will spend a good bit of time later in the semester talking about misfolded proteins, right? This is very important. Gives you, actually, I think about misfolded proteins as probably one of the like core central concepts to understanding, neurodegenerative disorders and such, right? I think that's going to be where the unlock is. Here you go. Here's your basic. We've all heard the slightly incorrect but still accepted central dogma of DNA, RNA, protein, right? You don't need one of those. Guess which one? It's the DNA, right? You can go RNA to protein. That's fine. Whatever. Whenever possible, I point out when Francis Crick was incorrect. It only happens twice, and this was one of them. We've got one more later in the semester. We'll talk about that. Oh, hey. How many of you love that guy who was in that movie, but you don't remember his name? But he seemed pretty like believable as like some second-tier character, right? Okay. His name was Astrocyte. We're going to learn about the glial cells. Okay. Glia comes from the word glue. Okay. Guess what Elmer's glue is made of? Not glial cells. It's other stuff. Mostly the same stuff you put in pasta. Pasta and paste are like the same word. You guys think about these things? Yeah. So next time you... What?
Is that why kids eat glue and stuff? Because it's basically just starch and water.
There you go.
Different concentrations.
There you go. Next time you're... So I'm thinking about this, right? Like, for people who try to carb load, right? Comes in those little squeezable packets. Even comes in different colors now. So much fun. See, brilliant ideas. You guys, I should get a patent officer to come in here and just write these things down as they come out of my brain.
This is literally your shower thoughts, isn't it?
It is. But it's important for you to think about them, right?
You also need to write that book.
Yeah.
You've been writing for years, decades.
Glue. Why did they call it glue? They thought it was actually holding your brain together. That's wrong. But all the same, not physically, but metaphorically it sort of is. So first we have astrocytes. We'll talk about these guys. These are awesome. Who remembers the... Oh, we didn't talk about the blood-brain barrier in this class, did we? No. We need to talk about the blood-brain barrier at some point. So now seems like an okay time to do it. The blood-brain barrier is a barrier between the blood and your brain. Just in case you didn't get that from the name. The point of the blood-brain barrier is, how many of you have ever ingested something you probably shouldn't have had in your brain? What is today? Tuesday? So if we go back like 5 days, is that Thursday? Then probably, right? I don't know if that's 5 days. Happens all the time, okay? Some of those things are very tricky, and they can get through this blood-brain barrier. We'll talk about that in a moment. Some things cannot get in the blood-brain barrier, right? You have this to prevent toxic substances from getting into your brain, okay? That's the point of the blood-brain barrier. Now, it's not just a single thing, it's multiple things. Largely, it's related to the structure of the capillaries in your brain. Most capillaries are full of holes, right? They're kind of like a salt shaker because stuff needs to come out pretty easily, right? You want it to come out easily so you can supply nutrients to the rest of your body. You don't want things to come out easily in your brain because they will come out and they will cause damage. And remember, we said damage in the central nervous system, you can fix it all the time, right? No, you can never fix it, okay? So you want to protect that. There are a few weak places in the blood-brain barrier. How many of you love projectile vomiting? Yeah, there you go, right? So the area postrema is responsible for projectile vomiting. If you eat something that's toxic, you start vomiting, okay? Because your blood can get into that brain region and it goes, whoa, that doesn't belong here. If it's in the stomach, I know how to get rid of it. And you just start vomiting. Astrocytes are great. They do a lot of cool stuff. We'll talk about that. We're going to talk about oligodendrocytes and their cousins, the Schwann cells. Okay. Schwann cells are in the peripheral nervous system. This is very important, right? That there are some differences between these two types of cells. We'll talk about that. Microglia are awesome. How many of you love brain inflammation? Nobody does. That's fine. How many of you love brain parasites? You want like a brain infection, right? Everybody wants one. Microglia are a part of the immune system that works in your brain, right? They're going to help sort of attack those invaders, right? That's like, remember, anybody use those neti pots? Have you ever tried one of those? Yeah. So it was like, those were like really popular for a while, and then I feel like they're kind of like down here now. Right? Like some people still use them. I'm not saying you shouldn't use them, but I feel like it was like this big craze because like Dr. Oz was like, man, pour water up your nose.
And then that one guy got a brain parasite, and everybody was like, maybe it's not such a good idea.
That happened right here.
Yeah.
That's why they tell you to use distilled water. Normally, you don't want to put distilled water in your body. Okay? You don't want to like go. Don't be drinking distilled water. You've been doing that, right? Don't do it. It's a bad idea. And don't give your babies, if you have them, distilled water. That's a bad idea too. Some people use distilled water. I don't know why. Distilled water, there's one place it does go, in your car. Right? That's a good place to put distilled water. Okay? Don't put it in your body. Yeah, but the guy used just like tap water. If you use the tap water here, you're taking a chance anyway, if you ask me. I double filter and then I boil and I distill it and add in some electrolytes. I don't know what I do. All right, let's talk about astrocytes a little bit. They're called astrocytes because they look like a star. Basically, they help reach out to the blood cells. They bring in some nutrients, right? Glucose in particular is awesome. How many of you love glucose? Everybody does, right? Your brain runs exclusively on glucose. That's why Fun Dip is so awesome, right? this was not a problem, right? Like things, this is one reason that we have such an issue with some dietary-related health problems, right? When the sweetest thing you could get your hand on was an apple, you could eat apples all day long and you'd be fine, right? Okay? Nobody's teeth are going to rot out because they have too much sugar. Nobody's going to get diabetes from eating too many apples. It doesn't happen. You can get these problems by eating these other really sugary foods, right? So when we have those, you know, we can pack sugar into stuff. We love sugar. We were evolved to love sugar. When all that was available to us was fruit on the vine, that's fine, right? It doesn't matter. You can just eat as much of it as you get your hands on. You don't know when you're going to get your next batch. You're going to be fine. You keep moving along. It's not a problem. But when you can go to wherever it is you get your powdered candies and buy cases, right, of just purple sugar, it starts to become a health issue. The same thing is true, actually, of other foods. You know, when you start having these calorie-dense foods, you have really high-fat foods. We love fat. Why do we love fat? There's so much energy in fat, right? And when you used to have to, like, run down your, like, literally run down your food because it was running, it didn't really matter how much fat was in it, right? When you mean, I'm going to go run down some dinner, and you're just, like, driving around to get a Big Mac, it's not really the same, right? So they just put Fun Dip on your Big Mac. It's a brilliant idea. You'd sell those things, just fly off the shelves. All right, so we're going to suck in some glucose. We're going to turn that into usable things, send that out to your neurons because they're constantly going to be doing stuff, hopefully, right? Especially those neurons that like make you breathe. You always want those turned on. Okay? So those guys need to be working all the time. Questions about this? Great.
About what kind of, like, if you have low blood sugar, you kind of get like a brain fog? Is that kind of related?
There's some other things involved, but definitely having low, if your blood sugar is too low, your brain's not going to work right? Because you're not going to have the glucose that you need. And you're going to start pulling glucose from other places, which we'll try to do. And that may not work so well. Sort of depends on your ability to do that. So you should always have like a packet of Fun Dip ready. Like, right? Like when you're in the middle of class and you're like, oh, wait a minute. I'm not getting this. Casey, do you not tell your students this stuff? Not about the Fun Dip. Jeez. I think it's getting hotter in here. All right. I guess we should talk about oligodendrocytes. These guys are awesome. Another glial cell, right? It's like a whole class of cells. Basically, you're either a neuron or you're a glial cell, right? I mean, that's it. Okay, so we're glial cells. Your oligodendrocyte will reach out to multiple cells, multiple axons, and it will make these sort of lipid-based chunks of insulation called the myelin sheath, right? When all of this together is called the myelin sheath. These are myelinated axons. Not all of your axons are going to be myelinated. Okay, there's some that aren't. Most are, right? Because we want that fast, high-quality signal there. Sometimes where unmyelinated axons are fine, we're going to do that. This is costly, right? Everything that you do in your brain costs a lot, okay? So we want to be very particular about what we do. We want to make sure we're only putting in the things that we're going to use. If we're not going to use it, we're not going to have it, okay? Here are your microtubules running down those axons. That's awesome. Next to that are the, this was your cue, mitochondria, powerhouse of the cell. We were waiting on that. That's where those guys are because we need those. We need that ATP to keep this. Because we're going to have ion channels here. They're going to be doing stuff at your nodes of Ranvier. What happens if one of these guys dies? Guess what? You lose some of this myelin sheath, right? Wherever it's reached out to. And wherever it's just reached out, wrapped around, you're going to lose that. What's that called? Somebody's thinking. Yeah. Multiple sclerosis, MS. Right? MS is a degenerative disorder. Right? It's going to selectively influence and affect your oligodendrocytes. Right? This is why you have lesions. Right? Some folks will say they have like lesions in their brain. When you look at the brain of someone with MS, they're going to have holes, basically. Holes where they used to have myelination. Okay? That myelination is going to disappear. It only affects the central nervous system. Right? Remember we said in the peripheral nervous system, we have Schwann cells. Schwann cells perform the same job. Okay? They myelinate axons in your peripheral nervous system only. Okay? They have a slightly different chemical composition, so they're not affected in MS. Okay? So that's interesting. On top of this is the real kicker, Morgan. This is exciting. Remember that time I said, if there's damage in your central nervous system, forget it. If there's damage in your peripheral nervous system, you can have some recovery. Okay? And that can happen because of your Schwann cells and their capabilities. They can regenerate and help the regeneration of axons. Okay? So they can do that. Can't do that in the central nervous system, but can do it in the peripheral nervous system. So if you have peripheral nerve damage, sometimes you can experience some recovery, right? Not going to happen in the central nervous system. Questions about that? That's interesting, right? Hey, guess who already talked about the blood-brain barrier? Again, that was us, in case you missed it. Here's some other details about the blood-brain barrier. Everywhere else you actually have these gaps, right? In the cells of your capillaries. Again, that's to allow the nutrients to flow in and out. What also flows in and out? Like toxic substances, right? Just anything. We're going to seal those guys up, okay, in the brain. We're just going to make these guys nice and snug so nothing can get out. Not a big deal. Here's another way you can organize neurons. We talk about multipolar, bipolar, and unipolar. Largely, I think that's useless. Okay? I think it's a useless division, and I think it's confusing. I've been studying brain cells for a decade, and I'm going to tell you, if you showed me a picture of one, I couldn't tell you what kind it was, looking at a multipolar, bipolar, or a unipolar. Doesn't make any sense to me, and I still don't understand the difference between a multipolar and a bipolar, or how something can only be unipolar. It just doesn't make sense to me, so don't spend your brain on it. What you do need to get out of this, there are different types of neurons based on their structure. This is the structure, the shape of the neuron. Okay? It's going to be shaped in a way that will allow it to perform its job. That's all you need to know. Okay? Neurons in the retina, right? Cells in the retina are going to be structured differently than cells in the cerebellum. They're doing two different jobs. We'll talk about those, so don't worry about it. So they have to be structured differently, even though they're similar, right? They're going to have some of the same parts. You're still going to have an input part, calculation part, and an output part. Okay? Might look a little different, but all in all, it's going to be doing the same thing. All right? We could talk about reflexes if you want. I don't know that it's that helpful. Here's what happens.
Is he touching the arm? Oh.
This is, you didn't know this. This is the correct way to determine if your iron is ready to use for your clothes.
Oh.
Yeah, always hand right on it. I do this with my stove too. Like, can it boil water? And I just put my hand right there. Yeah, that's a great way to do it.
At least he's touching it with his hand.
Yeah.
That's because he already used the other
I
nine.
put my hand straight on it. And
Straight on.
like, oh my god, I proved it. Dumb. What am I going to do?
Yeah, it happens. Maybe she was the one mom wanting the bigger head.
Maybe. Well, no, I was a breakout baby. So
Oh, yeah, there you go.
it didn't really matter how big my head was.
Sometimes it's the shoulders, not the head, that gets stuck. Yeah. And then your head comes out shaped funny. Because it's
Because they got
it's been in somewhere for a while that heads don't belong.
shaped heads.
That is the best way to eliminate people with funny shaped heads. Stop having kids. It's a surefire method. All right. A lot of reflexes. What's interesting about this is it doesn't have to go to your brain. That's kind of fun. Which is good because that saves you time, right? And sometimes even milliseconds can mean the difference between, like,
Second, third degree.
absolutely, right. This guy's still getting a burn,
Yeah.
but it may not be a third-degree burn, right? Which is great. There are sort of inhibitory neurons. You can override sometimes reflexes, right? That does go to your brain, and your brain can go, hey, wait a minute, I'm intentionally picking up something hot, right? Because I don't know, I just wanted some oatmeal. I don't know what this is. It looks nasty because it's like this dark brown steaming thing. All right. I want you to take a moment, not a long moment, but a short moment, and just go through everything we just said and put that over somewhere to the side. Now we're going to start over. And this is where it gets confusing. So hopefully nobody has been so lost up until that point they don't know what class they're in. At this point it may happen. Okay, and I just like to prepare you, scare you for a few moments so you'll actually pay attention going forward, right? Because up until now you may not have been doing this. I know some of you were. People who sit in the front rows. Those back row people. I never trust you guys. I used to sit in the back row, that's why I know. That's exactly why I know. So Casey, I met my wife in a psychology class, by the way, which is a miracle we did because I was always in the back and she was in the front. So, yeah, that was a fun fact, I guess.
I was wondering where you were going to go with that.
That was it. That's where I went. I went right there. Just left it. So far, I've told you all the pieces and what those pieces do. We've talked about the axon. We've talked about the dendrites. We talked about the soma. I alluded to this idea of electrical signals, right? Okay, I said, it's opening and closing these ion channels and sodium or whatever. Now we're going to really talk about this, right? And I think before we do that, we need to talk about some basic concepts. We need to get a few words and concepts under our belt so we kind of have an idea about what's going on here. What we want to do now is we want to actually measure electrical potentials. Okay? And what is an electrical potential, right? And this is kind of a vague concept you're going to get your head around. You can do this in an axon, dendrite, wherever, doesn't matter. We'll talk about it. You've got a cell. You've got an inside and you've got an outside, right? If you were to stick an electrode inside the cell and then one outside the cell, and you were to compare the difference in charge, electrical charge, between the two, that's your potential. Okay? How many of you have ever used like a multimeter? Yeah, okay, that's one person. That's the little box with a dial on it, and it's got a red and a black cord coming out with 2 points. Okay, it's not to roast hot dogs, in case you thought that. Pocket hot dog roaster, and that's kind of a fun thing. Turn the knob to done, I don't know. This is so you can measure like electrical currents, right? You may stick these into an outlet. Make sure you know what you're doing. Right? We'll be short a few students. Right? You can test batteries with these. Like see if that's a good battery, right? So it's a similar idea. We want to measure the difference between the charge outside the cell and the charge inside the cell. Okay? We want these charges to be different. Right? The reason we want them to be different is so then we can later make them the same. Right? That's how this works. So if you think about something, it's a lot like potential energy. How many of you are familiar with potential energy? You have like a book, up here. And let's say you wanted to, what's a fun thing you can do with a book up here? You can make an insect killer. This works. You got an ant problem? The physics book will take care of that. And the physics book will take care of that by just walking around and drop the book right on top. Okay? Works every time. It works better if you leave it in the cellophane, because you don't have to worry about the book opening on the way down. So it'll hit flat. Cleans up easy too. So we've got that potential energy, right? The difference between the floor and the location of your book. Okay? The higher you take your book, the more potential energy it has, right? The more forcefully it's going to hit. Okay? For our purposes, we're going to have the inside and the outside of the cell be charged differently. Later, because we're going to set up these charges with concentration gradients, right? With charges and chemicals on different sides, okay? With this electrochemical gradient. Later, we're going to open channels, and we're going to allow things to move back and forth, which is going to change that potential in a way that's going to send a discrete, and by discrete I don't mean a secret, I mean independent, right? Individual. It's going to send a discrete signal through that cell, okay? So there we go. For our purposes, I don't know what your book says. Anybody know what the resting membrane potential is in your book? I'll make sure I tell you that number. Is it negative 65, negative 70?
I always done negative 65.
Yeah,
But I
sometimes.
haven't read the book yet.
Some books will tell you negative 70. I don't remember what your book says. Negative 65 works. Doesn't really matter what it is. I mean, it does, but doesn't. We'll go with this. Inside the cell is going to be negative 65 millivolts. This is millivolts. This is small. Outside the cell, guess what that is? We made it really easy for you. It's 0. So we've got this set up. We'll talk about how we set this up in just a moment. Okay? But outside the cell, whole rest of your body, whole rest of your brain, we're going to say that's at 0. Neutral, not a big deal. Right there at 0. We're going to say the inside is negative 65, right? The point is going to be able to take that negative 65 and make that positive, right? We're going to change that briefly, right? So we have a positive spike, and then we can make a bunch of positive spikes, and if we make enough of those in the right pattern, we know we've just seen an apple. Okay, that's how this process works. Remember that time I told you every neuron is getting like 10,000 inputs at any given time. Some of these are yes votes and some of them are no votes, right? Okay. Some of them are going to be excitatory. Some of them are going to be inhibitory. We're going to constantly be moving this up and down just a little, right? Just a little. It's just going to be moving up and down. It's going to bounce around negative 65, right? But it's just going to do this all day long. Okay, up and down, up and down, up and down. Not a lot, just a little. If we go a lot up, then we've got an action potential. We'll talk about that. Here are some other words you need to know. These are confusing words. You don't have to write them down. You've got a book. You've got a YouTube subscription. You've got lecture slides. Notice I didn't say you had a brain to remember them, but you have all those other things you can go to. Okay. Depolarization is a reduction, a movement towards 0. Okay? This is going to be making something more positive. It's going to make it more likely to have an action potential. We're going to depolarize it, right? Because if we're negative, like if this is 0, and we're down here at negative 65, if we move towards 0, we are making it less negative, right? That's a depolarization. Okay? I think of donuts as depolarizing. Everybody loves donuts, right? Okay? Everybody loves donuts, depolarizing. You know what not everybody loves? Crepes. Crepes are polarizing. Some people like crepes, some people don't. So we talk about crepes. Javon, we've just polarized things. Okay? So we're going to move down here. Maybe it's negative 100 millivolts. Always label your units. I didn't do that. Okay. Reminds me of this great Mark Maron bit. I don't know if you know Mark Maron. Mark Maron, I think, is a great comedian. You guys should check him out. If you haven't seen, I don't know, I'll make a recommendation because you don't have anything else to do with your time. I loved the show Glow on Netflix. I don't know if anybody saw that. First episode, I almost died. I literally choked on some Chipotle at a joke that I can't repeat in class, but was hilarious. Casey, you should watch it. No one else.
That's about the wrestlers, right?
Yeah, it's for mature audiences only, I promise you. It's based on a true story, by the way. So for any of you who lived through the 90s, there was something called Glow, and it was like on a year or two. Frank Stallone's mother was on the show, which adds a fun wrinkle to... I know. That's Sylvester Stallone's brother, by the way, is Frank Stallone. It's a long story. There's a documentary, you should watch it. But he talked about like these people who were like on the other side of the like political realm from him, but he was like they both like Tom Petty, so he's like, but I don't know at this point if like Tom Petty's enough to keep us together, and so it was good. I would recommend that as well. Polarizing things, right? So it's going to make something more negative, right? In our purposes. We're not going to really think about, I know technically if we go really positive, that's hyperpolarizing, right? But we're not going to think about it in those terms, okay? Hyperpolarizing we're going to typically think about as making it more negative, okay? An action potential is a very, very brief, massive hyperpolarization. But we're not going to probably apply that same level or that same word or that same concept to it, okay? Those are the things we're thinking about. So depolarizing, hyperpolarizing. Depolarizing, hyperpolarizing, right? Constantly up and down. Doing that all day long. By the way, for those of you that want to know, this is called a graded potential. Why is it called a graded potential? Like this class, you could get an A, a B, a C, a D, F. I don't know what they had against E. You could have all of those things. So that's graded potential. An action potential is going to be something different because we're going to go along like this, and then all of a sudden we're just going to have that massive action potential. That's like a pass-fail class, right? It's going to happen or it's not going to happen. Okay, it's an all or none event, right? You guys have heard that. We'll talk about why it's all or none. I know, like sometimes they tell you like action potential is all or none, and it doesn't mean anything to you. And I'm going to tell you why that means something. But first, let's talk about diffusion, right? When you get in one of those situations and you're having a crepe battle with someone, and you're like, man, crepes are awesome, and they're like, no, crepes are not. And you're like, how are we going to bridge this rift between us? Diffusion, right? You got to no. So what's the student concentration like? Let's imagine it's not now. Let's imagine it's a Monday. Not a Mondays aren't good days. Wednesdays are better. Nobody comes to class on Mondays. It's Wednesday, and it is 10:40. No, 10:40 is not good. Nobody, because somebody might have been trying to get to class in. It's 10:30 a.m. on a Wednesday. How many other students are out there? 0. Do you know why? They're all in class until 10:50, right? Okay. So if you think about it, all the students are concentrated in a few places, right? And Harris Hall could be one of those, right? Okay, so they're all going to be concentrated in one place. At 10:50, completely different story, right? Instead of all of the, if you draw a map of campus, right, and you've got, I don't know, this is a really rudimentary map. Guess which one's Harris Hall? That one, yeah, that one. The really ugly square. Student concentration at 10:50 is going to be really high in all of these buildings, right? Or at 10:30. Okay? It's going to be really low, like on Buskirk Field, right? Okay? All you're going to have is that, like, one guy taking a nap in his hammock. Okay? I don't know. Is anybody that one guy who sleeps in the hammock? Nobody's going to admit to being that guy. So, that changes, right? So we've got all these guys concentrated. At 10:50, and we all say, all right, you guys can leave, class is over, right? Then, what happens to the concentration of students? Instead of being isolated, right, it all spreads out. It's sort of evenly, right? Because people are coming and going from buildings. up until almost 11, and then everybody's concentrated back into the buildings, and the campus is pretty clean, right? So this is why I plan my trips. Like, I'll always be on campus walking around at 10:30, but never at 10:50. Then I wait until like 11:10, because there's always a straggler coming through. Gotta wait on that guy to get off campus. Okay, that's diffusion, right? At 10:50, when everybody leaves their classes, we've opened a student channel, we've opened an ion channel, right? We've opened a door for you to go through, and then you do. That's how diffusion works. What drives diffusion? It's just this high concentration. There's also some electrostatic pressure. What's the first thing you learn about positive and negative charges? Like repels like, right? Okay, so if you've got a lot of positive charge in one place, a lot of negative charge in one place, they're going to like push against each other, right? There's this electrochemical gradient. I'll write that word down. Okay, electrochemical gradient. That's for you to remember. It's going to drive this. Diffusion is always going to be pushing, right? Pushing, pushing, pushing. With your cell, this is inside, this is outside. Outside that cell, you've really only got two things we want to think about. Sodium and chloride. You are basically, your brain is just a big ball of salt water with some cells floating around inside. It's all salt water on the outside. Super high concentration, sodium, lots of chloride. You're not going to run out of sodium. You're not going to run out of chloride. Okay? Not going to happen to you. I mean, I guess it could. It's not going to. So don't worry about it. Inside your cell, you got a few things. For our purposes right now, we want to think about potassium. And yes, that starts with a K. The K's silent. A good one. Also sodium. I guess I should have told you sodium. Na, that's Latin. Don't worry about it. So potassium, high concentration on the inside. You also have these organic anions. Okay, so you're going to have a high concentration of these organic anions. This is awesome. When we open channels, when we open a channel here, guess what happens? Sodium will rush in. If it's a sodium channel, potassium can go out if it's a potassium. Chloride can come in if it's a chloride channel. Organic anions can go nowhere. There aren't channels for those. Okay? They're going to be stuck on the inside. And this is brilliant. Because who remembers the resting membrane potential? You always want to have something negative in there, right? Because you're always going to be driving back to that negative resting membrane. We want to go back to negative. Why? Because we're going to start back to get that big positive spike again. Okay, so we always are trying to go back to that resting membrane potential. Are you ever at the resting membrane potential? No. You might accidentally pass it once, but you're not going to really be hanging out there at negative 65, right? So it's sort of it's a theoretical place where you can be. You're never really going to be there. If you all had all of your, guess what happens if all of your neurons go to negative 65 millivolts? Nothing. I mean, literally nothing happens because you don't have action potentials at that point, right? And all your brain cells just stop. And again, you want at least those ones that make you breathe and your heartbeat going all the time. The other ones, maybe turn those on and off, right? Okay. But you want to have everything else going. Now, one of the biggest, so we open these ion channels. Stuff's come in, stuff's come out. That's awesome. Here's the problem, though. What happens when we open the ion channels again? Well, we've already sort of bummed up diffusion, right? So that's not going to help us anymore because we don't really have that electrochemical gradient because we've let this go long enough that everything's evened out. So opening channels again is not probably going to help us a lot, right? What we do have, and this is the reason you love glucose. Remember that time I said you have to have glucose because your brain's constantly using ATP? It's constantly using ATP because of this guy called the sodium-potassium transporter. Okay, also known as the sodium-potassium ATPase, because guess what it does to ATP? Constantly chopping off that third phosphate, right? Yeah, it makes it ADP. Not the people who manage your paycheck. That's also ADP. Those of you that have a paycheck coming. I know, right? Also, it's called the sodium-potassium pump. Whatever, it's the same thing. Constantly using ATP, okay? And we're going to, guess what? Swap sodium for potassium. Now, most of you are thinking that doesn't get me anywhere, right? I don't know, right? I'm going to try to think of a good example of a trade here that might not get you anywhere. I don't know. What if you were to trade like LeBron James for Steph Curry? I don't know if that really gets you anywhere, right? Because you're like trading like two of the best players for each other, right? It might get you somewhere if you had a point like let's say you had like an extra point guard and you're like, I don't really need this guy. I got a guy who makes like 400 threes a year anyway. Why do I need a second one, right? And maybe Steph Curry becomes disposable. I don't know. Does that happen? I don't think it does. No. But I think about these sometimes, right? When you trade these guys, it's all about what are your needs, right? It's not about like, well, these are two awesome things. We know sodium's awesome and potassium's awesome, so why are we trading them? How does that help us get to that negative membrane potential? It helps us because every time we pump out sodium, we pump out 3 of those. And we pump in 2 potassiums. So if we're pumping out 3 positives and only putting in, right? It's not even 3 for 3. So we're pumping out more positive, so we're getting negative. It's 3 for 2. Don't worry about it. Talked about all this. I basically drew this for you, but here it is in a fun diagram without all my scribblings on it. Okay? This is your phospholipid bilayer. That's your cell. Remember, you've got this electrostatic pressure. We've got to open channels in here so these guys can flow in and out. Here's your sodium-potassium pump. 3 sodium out, 2 potassium in. It's going to create a net negative charge inside your cell. It's going to drive you as close as we can back to that negative 65 millivolts, right? That resting membrane potential. Okay. This guy, what it doesn't show you here, this is a total ATP drain. Okay, constantly using ATP. Every time it's pumping, it's slicing up an ATP, right? Okay, and you went through that Krebs cycle to make how many ATP? Something. Yeah, that's good enough. A good number, right? So you got to keep doing that. Okay. All right, who's still with me? A few people. That's your I'm lost alarm?
Yes, it is.
So far, we've done nothing. I know. So far, I have spent an hour telling you about how to do nothing. That's exactly what has happened, right? Because we've never made it past the resting membrane potential. We have yet to accomplish a single thing. We have not fired one single action potential. And you are going to have to fire, I don't know how many of those, right? Some number of action potentials at any given moment to accomplish anything, okay? Constantly going to be firing action potentials. So far, you've learned how to do nothing. And you thought you already knew how to do nothing. But here's like the real details of how to do nothing. Next, we need to talk about the action potential. When the action potential happens, and it's beautiful, it's probably the single most beautiful thing you'll ever see. I promise you. The cell, got that cell, remember that resting membrane potential. Our goal is to depolarize that, right? We've got to depolarize that. to, and this is the phrase, the threshold of excitation. Right? That sounds like you've reached nirvana, the threshold of excitation. Right? I've reached the threshold of excitation. This happens because we have moved really two ions. We've moved sodium and we've moved potassium. Where did we move the sodium? In. Because most of the sodium's out, so we got to move it in. What about the potassium? Right, it had to go out because most of it was already in, right? So that's where we made that flip. Okay. I know you're going to think, why am I letting the potassium out? There's a lot of things, we'll talk a little bit about that, because you want to think, well, I would want to hang on to that potassium, right? It makes it even more positive. But we've got a reason we need to get that potassium out. And one of those reasons is that sodium-potassium bump, so we can go back to that resting membrane potential. So again, we can still do nothing. This action potential, it's a brief electrical impulse. That's the words, right? This is the information that you're conducting along your axon, right? This is the information you're sending from one cell to another, okay? This is Johann Mueller's, doctrine of specific nerve energies, right? When he was talking about that electrical signal, that electrical impulse, he didn't know it at the time, but it was the action potential. The reason he didn't know it at the time is because Thomas Henry Huxley hadn't had a grandson yet who figured this out. All right, so threshold of excitation, that's just the membrane potential. That's where we reach the point of no return. Once we become that depolarized, right, it's going to happen. There's no way to stop it. How many of you have ever tried to tip something over that was heavy? Okay. So you're rocking it back and forth, right? This is a great way to do that. Rocking it back and forth. At some point, it's rocking so much, it's going to happen, right? So you just got to figure out which way is it going to go. That on top of you, right? And so you got to get out of the way, okay? That's that threshold of excitation. That's that point of no return for us, right? Once we get so positive, so many ion channels have opened that more ion channels are going to open, and so many ion channels are going to open that we're going to do an action potential whether you want to or not. You're on it, right? How many of you have ever been on a roller coaster? Okay, yeah. Threshold of excitation is when they release you at the top, right? Up until that point, things can happen. You can stop, right? Okay? Once you are at the top and they let you go, it's gravity from there. And there's nothing you can do but scream, vomit, and wet your pants until it stops. You guys ever been to Camden Park? Who's been there? Yeah, I can tell not many hands went up. The reason for that is, see all those empty seats? I know I'm not wrong. My great-grandmother rode the Big Dipper. I know I'm not wrong. That's impressive, isn't it?
I rode it this summer. It is still just as horrifying.
I know there are like 5 generations of my family have tempted fate on that thing.
Didn't somebody just die on it a couple months ago?
I don't know. I didn't mean to make fun of someone if they did.
I saw someone got stuck under the rattler. That's the
Really? Oh, wow. It's a...
It's a very,
Hey, who loves bell peppers? They look like bell peppers, don't they? We should talk a little bit about ion channels. We should talk a lot about ion channels, actually. We might even talk about pipe cleaners. Yeah, that's going to be exciting. So, ion channels. Who remembers the phospholipid bilayer? That's this guy. Things that can get through the phospholipid bilayer are what we call lipid soluble. Things that are lipid soluble are small, possibly. Small gases can get in there. How many of you love nitric oxide? Everybody loves that. Ask your dentist. Or your urologist, depending on what your problem is. Nobody thought that was funny. One of the main jobs of Viagra is to increase nitric oxide. It's a vasodilator. Other things that are lipid soluble don't have a charge. What do we know about an ion? By definition, it has a charge. That's what made it an ion. Guess what? It's not going to make it through the phospholipid bilayer unless you have a channel for it. And that's what these beautiful bell peppers are. Do they really look like that? Well, no. But it works for you. So we got an ion. It doesn't matter what the ion is. This can be any ion you want, although it's probably going to be sodium. It's the way this works. Okay? This ion channel, we got to open it. We don't want it to just open randomly, right? We want it to open it when we want so we can fire that action potential. We're going to talk about how that happens. It happens through the use of neurotransmitters, right? Yeah, that's the way we're going to think about this. So we'll open and close this through neurotransmitters. There will be like docking sites and all kinds of things on there. You don't need to know about that right now. We're going to add that later.
Did you say lipids go through the ion channel?
No. No. Ions go through the ion channels. They can't go through the lipids because they have a charge. So they're going to be repelled anytime they try to go through there. They're just going to bounce right off. Just like that. They have to go through the ion channel. And that, in or out. Depends on which way it is. Now, who's ready for the pipe cleaner story? By the way, pipe cleaners are actually used to clean pipes. I don't know if you guys know this. I know everybody's like, oh, they're just for like craft projects, right? But they actually clean pipes. Which makes me wonder if you have to be like 18 to buy pipe cleaners. But you don't, do you?
No. You can walk into the hot log now.
Yeah, that's Hobby Lobby for you, though. No comment there. The tobacco, not the cleaner.
The voting thing.
Yeah. No, that's why I was asking.
Ridiculous. I mean, like 7-year-olds would have
Here's the question for you. If I just open a hole, how do I know that only my special ion is going to go through that channel? Okay, well first, some ions have different sizes. And that happens. So maybe I open a really big hole for big ions, but then those little ones could slide through there too, right? So Cameron, that doesn't help us any. That's why we have to start thinking about pipe cleaners. We're going to think about pipe cleaners now, and then we'll think about them again later. I like to think of pipe cleaners when I think about protein structure. Okay? I like to use them as a great analogy. So there are basically 4 steps to protein structure. The primary thing we want to think about is the string of amino acids, right? So how many of you know about amino acids? These are the guys that build proteins, right? So we put these guys together, we make amino acids. That's your primary structure. Secondary structure, you start thinking about alpha helices. That was supposed to be an alpha. And my Greek letters aren't that great, your beta pleated sheets, okay? These are sort of secondary structures, right? Once you get these amino acids together, a number of them actually have charges, and those charges will attract or repel, and then they just sort of, like origami, they'll fold. The alpha helices and your beta pleated sheets will also sometimes have charges on them, and then they can fold and fit together. That's going to create your tertiary structure, right? Okay, so you're going to kind of fit these pieces together. And then your quaternary structure, that's when you take subunits, right? So you're going to take these other pieces and they're going to interlock. You create this beautiful channel. Now, the inside of that channel, this guy, is going to be lined with positive or negatively charged amino acids, right? And the structure and the shape of that channel and the pattern of those charges is going to determine which ions can fit in and out, okay? And that's how we're going to make specific ion channels for specific ions. Now, some folks have what's called a channelopathy. Okay, this often happens, we think about this more with your calcium channels, with your CAV channels, voltage-gated calcium channels. Where is one other place where you have voltage-gated calcium channels? Anybody know? Your heart, in particular, that one muscle. that you definitely need to have working to keep you alive, your heart, right? And so some folks have channelopathies where they're calcium, and they'll have a lot of cardiovascular issues. Often, certain mutations in your CAV channels will result in non-viability, right? So you don't want to mess around with those. That's exciting, right? You guys are learning so much today. Or at least I'm telling you so much. This may be the single most important slide in this entire course. Okay? This slide summarizes pretty much everything you need to know about an action potential. Right? It's right here. We've got a diagram, we've got the graph, we've got words, whatever you need, it's right here. Okay? This is the process. And it looks like it takes a long time, but really it does. It takes like a whole 2 milliseconds. So let's walk through this process with the action potential. Before we do that, let's go back to where we were like 10 minutes ago when we were doing nothing. And when we were doing nothing, we were doing this. Remember? Up and down, up and down. We were getting yes notes, no votes, excitatory, inhibitory stuff, right? And we were coming into that resting membrane potential. We were just hovering around that. I know here it says -70, whatever. It doesn't really matter, okay? Once you have reached this threshold of excitation, once you have had enough sodium channels open so that enough positive charge has entered the cell, there's nothing you can do but have an action potential. That's it. It's the end of the story. Class is over. No. There's a lot more to it. Your sodium channel's open, you're going to get up to this threshold of excitation. Now, what is threshold of excitation? It's small. Huh?
Is it the point of no return?
The point of no return. We're really talking about a change in maybe 10 millivolts here, right? It's a small distance. Now, you don't want to be too far away. Think about the guy who's too far away, right? Think about the guy whose resting membrane potential and his threshold of excitation are 30 millivolts apart. You know what that guy did? He died. How did he die? Well, he didn't do anything. He didn't do anything at all. He probably never even started breathing, right? Because that's too far away for you to get that much excitation. Okay, so you want to be right there on the cusp. Now, you don't want to be too close. What happened to the guy who was constantly firing action potentials?
He had a seizure.
He did have a seizure. And that's all a seizure is. It's just uncontrolled action potentials. Right? We'll talk about seizures later and whether they're local or generalized. That's something for you to think about weeks from now. We want to have control on this. We only want an action potential when we want it. When we don't want an action potential, that's fine. Okay? We want these guys to be brief. How many of you know Morse code? How many of you know Morse code exists? Yeah, okay. And you know it's just a bunch of dots and dashes, right? And you know like SOS, right? Like dot, 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 dash, 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 dot, dot, dot. I think that's right. Right? What if Morse code was just, there weren't like independent spaces, right? Like in between, and you just went, beep, like that was your code, right? Beep. And you don't know what you said there, right? You just like smashed a bunch of letters together. Action potentials could be like that. What if you just got excited and just stayed up there and went down? Well, it could have been a lot of things, right? When we have these little independent things in between, right? Then we know, oh, well, we have different spikes, and it's a code. So, we want to go up, we want to come back down. We want to go up again and back down. We want to do this rapidly. Okay. First thing we want to do is we're going to open sodium channels. Okay? That's going to drive our membrane potential up. We're going to depolarize, right? Okay, we're going to become more positive. Okay? Once we sort of have been creeping up a bit, and I say creeping, but we've been really zipping up, right? Because we're talking about this whole thing's like 2 milliseconds. We've been zipping up. Then we're going to have potassium channels start to leak out. This is called a leak. Okay? Remember, we need these guys to leak out, right? We need them to leak out so later we can reset this membrane potential, right? So we can have control on this. Okay? How many of you like to take hot showers? How many of you like to mix cold water in with your hot water? Yeah, the reason for that is that you all do, whether you believe it or not. Because if you just had straight hot water coming at you, your face would peel off. Right? So you have some cold water in there too. Right? Sarah, you look a little disappointed that it's not just like straight hot water coming out of your faucet for some reason. You had this like look of disappointment. No, you're fine. Okay. So we're going to temper that a little bit, right? So we've got this out so we can take care of that later. Once we reach, now we're up to maybe positive 40 millivolts, fine. Once we reach this peak, sodium channels, they only work for a brief period of time, right? And they're going to close. And they're going to stay closed for some period of time. It's going to make sense here in a moment why this happens. We call this the refractory period. And there are actually two types of refractory period. We've got the absolute and the relative. Okay? Once that sodium channel's been opened for a little bit, it closes. It's going to stay closed. So positive's not coming in anymore. But guess what was happening? Remember that potassium that was leaking out? Because the potassium's leaking out, our membrane potential starts to drop, right? Okay, so we're going to go back past 0, down toward negative. We briefly overshoot, right, resting membrane potential. We call this the after hyperpolarization. Okay, not too big of a deal. Then we're going to, through those normal mechanisms, we're going to come right back up to that resting membrane potential, and we're going to hang out there until something else comes along and says, hey, it's time you fired an action potential. You've not done that in a good half a millisecond. So do it again. Let's go back and think about those sodium channels a bit, right? And how awesome they are that they have that refractory period. I think about this, I think about sodium channels kind of like football players, right? Because they'll run a play, like how long is a football play? That's like seconds. And then what do they do after that? Just hang out. And then they waddle up to the huddle. They got to bend over for a little. I'm not making fun of football players. I'm not saying it's not difficult. Right? Got to bend over for a little bit. Some guys got to yell out some random numbers and words. They're like, all right, let's go back and do this again. And then they go back and they do it again, right? Now, then I like to think about Peyton Manning when I think about this. When I start to think about refractory periods. And the reason for this is Peyton Manning was brilliant at the 2-minute drill, right? He had probably one of the most efficient 2-minute drills in the history of football, right? So he could get his guys, and they could run as many plays in 2 minutes as you could run an entire half of football, right? Because they were just that efficient. So I think about that. That's the relatively refractory period, right? So he's going to get these guys, and he's going to like motivate them just a little bit extra to squeeze in an extra play, right? And that's what you would do in this relative refractory period, okay? There's always an absolute refractory period, right? And if you watch football, what's the absolute refractory period? That's the chain game, right? Because it takes them time to move the chains and the referee to spot the ball, and so you got to start again. So there's always going to be, right? It's not like hockey. How many of you have ever seen a play in hockey? They run plays, believe it or not, but you don't know, right? It's constantly moving around, right? There's no discrete units in hockey. It just happens. They don't even stop the clock for people to come in and out of the game. They just like storm the walls, and then somebody jumps back in. Huh?
It's a perfect time to mess up.
I know, right? And then somehow the referee can count that fast and go like, man, you had 7 guys out here, sorry. There's a penalty. Somebody's got to sit down for 2 minutes. I don't know, right? It's very fast-paced. There's not any discrete sort of units. Football has discrete units, right? We're constantly play. You can do that. You can measure this was a play. You can say that. You can't say this was a play in hockey. This was just some sequence of the puck moving. And people clashed. So you've got your absolute refractory period. You're going to go down. Peyton Manning's going to extra motivation there. Back up. Okay? That's your ability to have extra action potentials ride on this. You're already up there, right? So if you want to fire more action potentials, great time to do it, right? You're already up there a little bit. Don't go all the way down. Provide a little extra stimulation and you can fire more action potentials. That's your relative refractory period. The absolute refractory period is that time when you can't, no matter how much you motivate them, you can't run another play, right? No matter how much your offensive line is ready, if the referee's not put the ball in front of the center, he can't snap it, right? So you've got to wait until that happens. Okay? Questions about the absolute or the relative refractory period? This is actually important. This is important because it prevents you from doing this, right? Just constant up and going. Okay, that's just confusing. And it prevents you from doing this. And then how much longer do you have to wait to fire another action potential? You can fire a lot of action potentials rapidly, right? Okay, so you can fire these clusters of these spikes. And that really adds extra sort of layers to the code, right? Because you could fire a bunch of action potentials and then stop, and a bunch of action potentials, right? And that means something.
Is it costing to do
Boy, is it costing. Yeah. You've got to really have a lot of stimulus. You've got to really have a lot of drive to make this happen. Okay, you're really, the stimulation level has to be really high to fire these extra action potentials.
I mean, on like long term.
Oh, no. Don't. No, you're not going to overthink yourself, okay? You're not going to think yourself to death. Unless you get up early like Descartes. As long as you sleep in, take a nap, you're going to be fine. That being said, I guess I should talk briefly about excitotoxic lesions. There is the possibility, right, in sort of a laboratory or a pathological setting where you could cause cell death, right? And one of those would be like a seizure situation if someone had a seizure disorder and they were constantly having these uncontrolled sort of action potentials and activity. That can cause some long-term damage. In the laboratory, you might pump in a chemical that would excite the cells to the point that they would die. Okay. But for the normal situation, this isn't going to happen. Okay, don't worry about it. I know you're like really concerned about brain health, and you were thinking maybe I shouldn't try as hard in this class because it might burn out a few things, but it's not. You're good. I promise. Hey, what's this? This is something. I guess this is when, let me see. I got to think about when we should talk about Hodgkin and Huxley, and maybe now's a good time. This is Alan Hodgkin and Andrew Huxley. We don't want to confuse him with his half-brother Aldous. We don't want to confuse him with his grandfather Thomas Henry Huxley or any of the other Huxleys. This guy won a Nobel Prize. Actually, both these guys did. That's the Nobel Prize medallion. Like horrible. It's not great, maybe. So these guys won a Nobel Prize. They actually... So this is one of the things that does irritate me about your book. They will tell you that they recorded from the giant squid axon. Most every book will tell you they recorded from the giant squid axon. They will proceed to tell you in another paragraph of explanation that it's not the giant squid from which they pulled an axon. The squid has a giant axon, right? So they will tell you this is a regular squid. It just has a really big axon. Now, why they just didn't tell you the first time it's the giant axon of the squid, like that just would save a lot of ink, right? I don't know. Maybe it was like formatting. They didn't have a diagram for that page. It was like, let's make this a little longer. Fit in the words. A squid has a huge axon. This is awesome because you can pull it out and you can stick things in it, and you didn't have to really have advanced technology. Now, we can stick stuff in axons like you wouldn't believe. I mean, you can just zap an axon from 50 meters with an electrode, and you're good to go. But, when these guys were doing it, not so much. So what they would do is they would stick in two electrodes, they actually were the first people to measure an action potential from an axon. That's pretty amazing, right? I mean, you guys could get a Nobel Prize doing that, right? I mean, you know, you can get squids. Jewel City probably sells them, right? Go get you a squid, pull out an axon.
I've never been there.
Really?
Yeah, I lived in Huntington 2 years now, but I've never been there.
You know, I used to eat there a lot.
Really?
Yeah, and then I got, I don't
Oh
know.
no, is it bad?
No. I mean, the food is good. I mean, it's the best seafood you'll get in town.
Okay. You're not going there.
It's like the only seafood you'll get in town.
Surprisingly, I still don't have a positive thing.
No. I mean, it didn't really. It's not necessarily great. I would eat their trout sometimes, and then I was like, so for like 5 seconds I was excited because I was like, man, that's like cool. They've got like local trout. Oh no. Yeah. And so then that one I was like, maybe not. So there you go. Their other stuff, obviously, like squid they have, that's not local.
Do they really?
Yet. 10 years, who knows where the ocean will be, but yeah. So I always think about that. Like, what's a good real estate investment? Like property here, right? That's on a mountain, because eventually the water is going to come up, and then it's beachfront. You guys got to plan ahead. You guys are going to live that long, maybe. So, Hodgkin Huxley, they recorded these action potentials. That was pretty awesome. They got the Nobel Prize for that. Now we actually have something called the Hodgkin Huxley model. So if you want to do a mathematical model of how an axon works, it's named after these guys. It's complicated for theoretical neuroscience, but not something you really need to worry about. Here's the cool thing about this. No matter where you measure this action potential down an axon, it's the same. It's not changing, right? It's the same size, same shape. It just is an action potential. That's what it was when it started. That's what it's going to be when it ends. They're all the same. An action potential is an action potential. Okay. That happens because who remembers those? That's a jelly roll. That's a myelin sheath, right? Okay. Remember the myelin sheath that goes around this axon that helps us with those action potentials. It's pretty awesome. This is kind of a basic idea. It's the rate law, whatever. I don't care if you know this. We've already talked about a stronger stimulus will elicit more action potentials than a weaker stimulus. That doesn't really tell you a whole lot about what the stimulus is, but it tells you about the strength of it. Don't worry too much. Here's saltatory conduction. This is actually kind of an interesting place to once again talk about sodium channels. Have you ever thought about how much of your life depends on a sodium channel? Yeah, the answer is all of it. 100% of your life depends on sodium channels. Never try to live without them. Who remembers that time I said sodium channels have a refractory period, right? There's that absolute refractory period. There's nothing you can do during that time to fire an action potential. This is great. The reason this is great is because if I take a piece of axon and I give you that piece of axon and I want you to tell me which way is toward the soma and which way is toward the terminals, you know what you're going to say? I don't know. And that's not just because you're inexperienced and don't know what you're doing. That's because there's no way to know, right? There's no way to know. This is not, it's not like there's a big road sign that says, this way to the terminal. Okay? Doesn't say that. It's all the same. Doesn't matter. So how do we know? Like you know what's really great? How many of you have ever used the mail? Right, how many of you pay one of your bills through the mail? One person does? Yeah, I don't know. You're behind. So either you don't pay your bills because your mom does, or which I think a number of you are in that category, or you use electronic bill pays, right? But it works the same way. What if you were sending an email and halfway to its destination it goes, I can't remember which way's forward and backward. I'm just going to go back to your inbox. That does you no good, right? It's the same thing with an action potential. If an action potential gets halfway to its destination and turns around, that was a waste of time. And it could have been a fatal waste of time. Like if you were trying to run from a bear and you were running and your legs were moving, right? So in case you think about it, I don't know if you've ever run from a bear before. Yeah, that's good. So you run toward the bear, right? So, It wouldn't be first inclination. But it surprised the bear. If you want that element of surprise, run toward it. You never know when you're going to catch the bear off guard.
It might shock it.
That's what I'm thinking, right? Just headbutt it right in the chest. So you're running from that bear, and you're telling your legs, contract, reach, contract, like all the time, right? And then all of a sudden that signal goes, I don't know which way I'm going. I'll go back up to the brain. And your legs go, and then you're dead. So that's bad. The reason that doesn't happen is because sodium channels and the sodium channels absolute refractory period. Okay? So there's a sodium channel, right, wherever on this axon. We're going to say here. That guy opens. Awesome. Sodium comes in. It closes. Sodium can't come in. Guess what happened to the guy behind it? He's still in his refractory period. So which way does this charge have to go? It's got to keep going forward, right? Okay? So that absolute refractory period lets us set up these axons so that everything has to move forward. It can't go in the reverse direction. Theoretically, and this happens experimentally, you could start the action potential down here. You could put in some kind of electrical charge, and it would go the other way. Right? Because of that absolute refractory period. Okay? So sodium channels are going to open and close, open and close, open and close. And because they're closed completely for a brief period of time, that charge can't go backwards. It's got to keep going forwards toward those terminals. So that's pretty cool. I think it's pretty exciting. All right. That's what's going on inside of a neuron. Now we've got to talk about two things, and we're going to talk about the two things simultaneously. Either, and it doesn't matter which one you think about, either how this neuron is telling another one what to do, or how it was told what to do. Works the same way, right? There's going to be communication between neurons. We call this synaptic transmission. The synapse is sort of that that's the cleft, technically. The synapse is sort of this all-encompassing area that includes the presynaptic terminal, the postsynaptic terminal, the proteins that are lining both of those membranes, the space in between where stuff's going to travel. That's all the synapse. Okay? We're going to send information across that synaptic cleft, right? So we can influence the other cell. So we've got the presynaptic cell, that's before the synaptic cleft. The postsynaptic cell, that's what's after it. So you've got, typically it's drawn like this. It's going to come down, and then you've got some other sort of like dendritic spine or whatever over here. Sort of this whole area is the synapse. This space in between is the synaptic cleft, right? Stuff's going to diffuse across there. If you've seen a Zoloft commercial, you've seen this, right? Because they have those little circles of things that just travel around and, make people happy. I think I've seen that commercial. What's going between here is neurotransmitters. We'll abbreviate that NT. There are a variety of neurotransmitters. You guys have probably heard of dopamine, acetylcholine, serotonin, norepinephrine, glutamate, GABA. Those are some of the big guys, right? We'll talk about those, maybe some others as we need to, when their particular systems come up. What's not going between here are ions. Okay? We are not sending ions from one cell to another. Okay? That's why we call this an electrochemical signal. We did the electro part, now we're going to do the chemical part. The chemical part, that's the neurotransmitters. When that action potential is coming down, right? When it's coming down that axon, it's going to get in your synapse here, your terminal. It's going to release these, we call them vesicles, synaptic vesicles. Inside of that are the neurotransmitter molecules. They'll make their way down, they'll dock here. At that presynaptic membrane, and they'll dump their contents out into the synaptic cleft, which will then be grabbed by the ion channels or on the other side, right? And then ions that are never been up in the sky, ions that are here can just come in and do their thing. We have this concept of a postsynaptic potential. When we're talking about potential, we're talking about changes in that electrical current, right? These could either be a depolarization or a hyperpolarization. Remember, we can open sodium channels or we can open chloride channels, right? It doesn't matter. We open chloride channels, a bunch of negative ions flood in. We're going to hyperpolarize that guy, and he's like, I'm not doing an action potential. Forget it. Here's a complicated concept. It's called a binding site. It's a site where something binds. I know. You've got your receptor. Why do they call it a receptor protein? Basically, everything's a protein, right? And the receptors are going to be proteins. Your ligand, what is a ligand? For our purposes, a ligand is going to be a neurotransmitter. Dopamine, that's a ligand. Anything that's going to bind and be able to operate that receptor, that's a ligand, okay? And everybody uses that lock and key analogy, and I think it's a little overplayed. We need a new one, but that one works perfectly well, so we'll go with it. So the neurotransmitter is going to bind to this binding site on that receptor protein. It's going to cause it to do something, right? It's going to open, let ions move in or out, whatever it's doing. We already talked about dendritic spines, presynaptic membrane. Postsynaptic membrane can be a lot of places, right? This is the guy that's going to receive the message. Could be an axon, could be a dendrite, right? Could be on the cell body. Doesn't matter. Here's a beautiful picture of, you know, if you're into 1990s turquoise and purple. I don't know. It's like Huntington Blizzard colors. Anybody remember the Blizzard? Yeah. Yeah, that was something for like 3 weeks, right? For those of you that don't know, this city had a, was it semi-pro or like minor league hockey? I don't know what the difference
I think
is.
it was minor league hockey because I used to come up and watch the games all the time. I think it was minor league.
Yeah, but it was like 3 weeks they had
Yeah.
a team. No, it was longer than that. It was like 2 seasons maybe?
Yeah, I think
2 to 3 years.
They were
There's
called the
a
Huntington
petition to
Blizzard.
Bring it back.
Really?
Yeah.
Yeah, that's going to go somewhere.
Yeah. Apparently there's a lot of.
I'll show you where it goes. It's going to go in the trash can because I don't think anybody's going to do anything with that. I really don't feel like the city's funds are going to go to bring a hockey team. You know, like the $8 they've got, they're probably using it for something else.
We can't even get our own trash or our own snow plows. So
I know, right? I
I
mean,
don't think we're gonna have a.
and they just cut back the recycling program.
Yep.
It's like, so we're not getting a hockey team.
We have bigger problems anyway. We get drug addicts running into corners. We have other things to fix.
Yeah.
Yeah.
You've got a synapse. You got trillions of synapses. You got trillions of synapses. Who knows how many of these things you have? You've got a lot. That's the correct number. That number's not going to change. Somebody says, Casey, how many synapses? You say a lot. And you just walk away because you know you're right. So we've got that action potential coming down. By the way, this is really hard to draw on the side. When it comes down this axon, it's going to cause some funky changes. We don't really need to worry about that. We're going to shake loose these synaptic vesicles, right? Okay, they're going to be docked in here, not to worry. When we shake those loose, they go down to this presynaptic membrane. They're going to merge with that. They make these beautiful structures. They dump all these little molecules out into that synaptic cleft. There's your postsynaptic membrane. You're going to have a bunch of proteins in here, in particular ion channels, right? These receptors, those molecules are going to bind. They open. Here's a molecule of sodium, just rushes right in. Okay? And guess what we've done? Well, we've affected the resting membrane potential, right? That membrane potential of that cell, making it creep up. If enough of these are opened, right, here and elsewhere, not just here, but elsewhere, guess what we have? We have an action potential, right? If we get enough of these to open, and we get enough sodium channels open, and enough of these sodium ions to go in, we're going to get that depolarization. We're going to get up to that threshold of excitation, action potential. We've passed on our signal. We're good to go. We're done. We can go back to resting membrane potential for the rest of our... No, because we probably got another signal we got to send. All right. Let's talk a little bit about... Wow, we've got like 12 minutes left, right? You think in 12 minutes I can get through 12 slides? That's like a minute a slide. I spend like 40 minutes on some slides. Let's talk about ionotropic receptors. There are two kinds of receptors. Well, there are like millions of kinds of receptors. There are two kinds of receptors, two categories. Ionotropic and metabotropic, right? These are interesting. So far, I've basically assumed that we were talking about ionotropic receptors. I didn't bother to tell you there was anything else. Okay? Here's how this works. You've got a closed channel. You've got a neurotransmitter that binds. It opens. An ion goes through it. It's a direct one-to-one relationship. Wherever the neurotransmitter is, that's where the channel is. That's where the ion is that comes through, right? It's right there. It's local. It's one-to-one. It's fast. It's fast on. It's fast off. We're done. Whenever the neurotransmitter disappears, the ion channels are going to be closed. And we're not going to be able to open them. That's going to be the end of the effect, right? We're done. That's the story I've been telling you. There's another story, though. Metabotropic. This is not a one-to-one. This involves G proteins. I know nobody loves G proteins, right? Second messengers, right? We've got a bunch of other stuff going on here. Here's the deal. You've got a receptor. When it gets activated, still the same story so far. Neurotransmitter binds to receptor. Okay, that's the story we've been telling you. It's actually going to activate the second messenger system that can go out and cause other ion channels, more than one, multiple ion channels to open, but it opens them from the inside, right? Now, this is a one-to-many. Okay? One-to-many. It's a bit slower. Now, we're not talking slow. It's just a bit slower, right? Because you got an extra step or two there, okay? And it's a cascade, right? So, Casey, recently, my wife and I caught 5 feral cats. Yeah, that was exciting. And we had a few things removed from them. Yeah, depending on whatever they had, we got it so they're not going to have more of those. Because like 5 feral cats is enough. You don't want 50, right? And they also do a little on their ear, so you know, like, that guy's got no parts. And you don't have to try to do it again, right? Because they were, and so we had to let them loose. So if you have like 1 cat, that's something, right? But 1 cat can have like 3 cats. And you know what 3 cats can have? 9 cats.
That's when you have a population.
Yeah. And then those 9 cats have... Right? This is what happens with these receptors, right? Because we can activate multiple ion channels with just one molecule, right? So we can have a wider effect. It can be broader effect. It takes a little longer. It lasts a little longer, right? Because you're coming in from the inside. And it doesn't have to be right there, right? So this, I mean, now this second messenger system, it's not going to travel like across the country, but it can travel a little bit of distance, right? So there's some distance there, okay? So it's not as spatially coupled, right? So there's a little bit of space. So there are some differences in those two types of receptors. All right. Most, a lot of your neurotransmitters will be able to operate both kinds of receptors. Sort of depends, right? Depends on the system. We'll talk about those as we need to. But when I say, hey, that's a, metabotropic or an ionotropic receptor for acetylcholine, then you need to think about what does that mean, okay? So you have to have that in the back of your mind. We already talked about vesicles. Don't worry about release zone. This is the zone where we're going to release neurotransmitters. I mean, I know, right? Jakia, I'm just throwing out these terms in there. It's like, what is a release zone? That's right there. Postsynaptic potentials. We talked about that, right? So when that opens an ion channel on that postsynaptic membrane, it's going to cause up or down movement of the membrane potential. Guess what? If it's excitatory, we call that an EPSP, excitatory postsynaptic potential. That's going to be the opening of sodium channels. If it's an inhibitory postsynaptic potential, IPSP, we're going to talk about chloride channels. Not a big deal. I don't know why the word reuptake is here. It doesn't need to be, because it's on this next slide. If we want to eliminate and stop the effects of those neurotransmitters, how do we do that? Okay, we can do it in a couple ways. One of those is called reuptake. Okay, how many of you ever heard of an SSRI? I can't spell SSRI. It's right there, right? That's a selective serotonin reuptake inhibitor, right? So we're going to block the reuptake of serotonin. Now, when we block that reuptake, what does that mean? That means that we're not, normally what you do is you release serotonin, you leave it out there for a little bit, and you're going to just pull it back. Okay? That's great. That's going to end the effect of serotonin. If for some reason you don't have enough serotonin, right, and we'll talk about agonists, antagonists later in a lot of detail, you want to block that reuptake so your serotonin stays out there longer and it does more for you, right? Okay, so that's how like antidepressants sort of work. So reuptake blockers, those are awesome. That'll work. Enzymatic deactivation. Now that sounds really complicated, especially when I tell you it happens through acetylcholinesterase. I know it's a long word, right? Just think if you knew that in the 3rd grade, you'd all be spelling bee champions. I lost a spelling bee in the 3rd grade once. Believe. That was the word that got me. I know, right? Of all the words, believe. I still don't know how to spell it. Doesn't matter. I mean, so acetylcholinesterase, or abbreviated ACHE, this guy comes in and it takes acetylcholine, right, and chops it in half. And so you get an acetyl ion and you get choline, right? It's not a big deal. So we're going to be busting up acetylcholine. That's going to stop it because it's no longer acetylcholine. So guess what it's not going to do? It's not going to work acetylcholine receptors, right? Because it's not acetylcholine. So that's awesome. And why that's awesome, how many of you have ever, and I know you all have, because I can tell, you've ever like, I'm going to flex my arm, I'm going to really, that required some acetylcholine. You're really lucky, because acetylcholine operates your muscles, right? It's what causes your muscles to work. You're really lucky that you have acetylcholinesterase. If not, you'd be walking around like this the rest of your life, because you wouldn't be able to unclench your muscles, right? Because they're just going to stay contracted, which happens sometimes. And we can talk about that when we talk about agonists, antagonists, and different sorts of chemicals that affect acetylcholine, which is fun. As homework, everybody go home and put a Black Widow spider on your face. Nobody's going to do that. Hey, I'm feeling like we can get through this whole thing. We've already talked about this, right? We've already talked about, here's an ion. This is giving you specific examples. Sodium, potassium, chloride, and then this business here, that's a metabotropic. You've got calcium, operates this enzyme. We'll talk about some detailed examples there. Here's basic reuptake. You've got a guy that's a transporter. It's going to suck up whatever that is. You call that serotonin. If you want to get super fancy, you can abbreviate that 5-HT. If you block that reuptake process, you get more serotonin down here. You're less irritable. You're sleepy, but you're less irritable. Neural integration. This is not really anything to think about. We've already talked about this. Remember when I was telling you about, excitatory inputs, inhibitory inputs? You're going to do that calculation, and you're going to decide here at the axon hillock whether or not you're going to fire an action potential. If you get enough positive ions, you're going to go, woohoo, we're going to do it. If they're negative, you're like, I'm going to wait, see what happens. Autoreceptors, this is sort of an interesting process. We'll talk more about this with agonist and antagonist. Basically, it's a way that you can monitor how much of a neurotransmitter you've released, right? So if you dump out a bunch of dopamine, your autoreceptors are going to monitor that. And if they go, whoa, man, that's a lot of dopamine, they'll slow you down. And if they're like, wow, I'm not seeing any dopamine, keep dumping it, right? Other types of synapses, we talked about this. Dendrodendritic, axodendritic, axoaxonic, not a big deal. That's just an example. Oh, man. Do we really want to talk about hormones now? Probably not in detail. But know that hormones can have effects. We'll talk about hormones later. We'll spend a whole lot of time on that, which will be fun. Here's some more homework. Everybody take steroids. With the Black Widow venom. Let's see what happens. Steroid hormones, not a big deal. All right. Who has questions, comments, concerns?